Hello everyone. It's very glad to be here to present my research work. My topic today is humor exposure levels and the difference of PBDEs and dl-PCBs in a typical e-waste dismantling area. As we all know, the severe pollution in e-waste dismantling areas has gained a lot of attention over the past 20 years. And due to the addition of the BFRs in the printed circuit boards and plastics of electronic products, improper disposal of direct incineration of e-waste can release and produce a large amount of pollutants such as POPs. The pollution problems of POPs such as PBDEs and PCBs in e-waste dismantling areas are very severe. And many research found that the concentrations of PBDEs and PCBs are at relatively high levels in various environmental metrics in and around e-waste dismantling areas. And the potential health risk to human posed by PBDEs and other pollutants and e-waste dismantling sites or areas have gained worldwide concern. However, there has been no systematic studies of the exposure differences of pollutants such as PBDEs among different populations. As we know here, can serve as a frequently used biomarker in studies of the environmental exposure to pollutants because they have stable. They are the hair is stable biological metrics, and it is easy to collect and preserve. And the sampling process is non-invasive, and it reflects a longer time window. Many studies have shown that there was a significant correlation between human hair and individual PBDEs in corresponding human serum, breast milk, and placenta. The concentration of PBDEs in indoor dust was positively correlated with the concentration of PBDEs in residents' hair. So we decided to do a little research about the the hair in the as the biomarker in residents from e-waste dismantling area.
Hello, Dr. Li.
Okay, sorry. Can you hear me?
Please share your PPT. We cannot see your PPT.
Okay, sorry. Is that okay?
It's just it's the first page.
Okay, sorry. Let me. Okay now?
Okay.
Yes. Okay. Now let's move to this page. I'm just talk about the sampling. We conducted the sampling in 2020 during the COVID-19. So we just collected limited samples. The residents of the volunteers from Guiyu, Guangdong Province, China, and they are all non-occupational exposure people, and their age range from to 44 years old, and we have 8 children and 8 adults, 8 males and 8 females, and some of the residents have been moving away for years. This table, it shows the details of the information of these volunteers. Next, we did the sample analysis. We washed the hair samples by milking water for 3 times and dry them and cut it to 2 to 3 millimeter. And then we used the succinate extraction, and after purification, we used the HRGC HR mass to do the determination. At the end, we detected 8 PBDEs and 12 dl-PCBs from the hair samples. Let's see the results at discussion. This figure shows the concentrations of PBDEs and dl-PCBs in the hair samples, and the total concentrations of PBDEs range from 60.7 to 77.8 nanogram per gram, with an average concentration of 22.3 nanogram per gram and a median concentration of 70 nanogram per gram. And the total concentrations of the dl-PCB were rather lower than the PBDEs, and they ranged from 79.1 to 901 picogram per gram. And the WHO TQ concentration of dl-PCB ranged from 0.04 to 2.98 picogram per gram. And the concentration of PBDEs in the human hair from this of the volunteers were relatively lower than these people from the US recycling workers, as they are the occupational exposure. The concentrations of PBDEs and dl-PCBs in here varied among different age groups. For PBDEs, the concentration levels of PBDEs in the hair of preschool children, school-age children, and adults were relatively similar, with school-age children have relatively higher levels of PBDEs in their hair. For dl-PCBs, the concentration levels in the hair of preschool children, school-age children, and adults generally showed a trend of gradually increasing trend. And the changes in the living environment significantly affected the concentrations of PBDEs and dl-PCBs in human hair. The levels of PBDEs and dl-PCBs in the hair of people who continuously live in the area were generally higher than those who have left the area. We can see it from this figure that the hair from the people who have left the area are generally lower than those stay in the area. And for PCBs, they have similar characteristics. And the concentrations of PBDEs and dl-PCBs in human hair show the gender differences. We can see it from those two figures, and they show that the average and median concentrations of PBDEs and dl-PCBs in women's hair were both slightly higher than those in males. And for the cardinal characteristics, we can see from that cardinal concentrations of PBDEs also show gender difference. The distribution of the PBD cardinals in the hair of males and females was slightly different, with PBDE 209 still being the most predominant cardinal in the hair of both populations. However, the levels of BDE 209, BDE 28, BDE 47, and BDE 99 in women's hair were significantly higher than those in men's hair. And the gender difference in the concentrations of the individual PCB cardinals was not significant. The concentrations of PBD cardinals in children and adults hair show slight differences, and we can see them that the BDE 209 remained the predominant cardinal in the hair of all populations, but the concentrations of lower brominated PBD cardinals, such as BDE 28, BDE 47, and BDE 99 in children's hair were significantly higher than those in adults' hair. The concentrations of dl-PCB cardinals in children and adults hair also show differences. The concentrations of PCB 77, PCB 105, and PCB 118 in adults hair were slightly higher than those in children's hair. We also found that there was a certain correlation between PBDEs and dl-PCBs in the hair of different groups. It indicated that the difference in the main source of the pollutants in the hair samples of these groups. We also did the PCA analysis, and the PCA analysis results shows that the samples from S3 to S9, which are within the blue ellipse, come from teachers and students of the local school. Except for S1 and S2, the samples within the red ellipse are from residents who have left the areas for several years. So this PCA analysis results also show the different pollution sources. That reflect the different pollution sources. Let's come to the conclusion. In this study, we found the concentrations of PBD and dl-PCB in the hair of the non-occupational exposed residents in U.S. dismantling areas were generally lower than those in occupational exposed populations, and were close to the concentration levels found in the hair of ordinary urban residents. And the concentrations and the cardinal distributions of PBDEs, dl-PCBs in residents here showed different characteristics because of different age group, genders, and those who have experienced the changes in their living environments. That's all. Thank you very much. Thank you very much.